Welcome, PCVs. I'm Elizabeth Farnsworth, Senior Research Ecologist with New England Wildflower Society, and I really want to thank you for your volunteer efforts. We're out in the field here today to show you how to count plants. We're going to use a common plant rather than a rare plant as an example of some of the challenges that you might encounter in the field in terms of trying to figure out exactly how many plants you've got. So we're going to start with this blackberry here, pretending that it's a rare plant, and I'm going to just simply show you what I use as a technique for counting individual plants. And it's really pretty simple. These plants are pretty distinctive, and I can count them each of the stems separately. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and so forth. Now, sometimes you bump into a situation when you're not actually certain whether each of these separate stems that you're counting are actually separate plants, because many plants are colonial or have rhizomes or runners that attach one stem to the other. And this is what we would call ramets. Those individual stems that you're counting are ramets that are part and parcel of one genetic unit. They're all identical. And that genetic unit we call a genet. Now, I typically try and count ramets as separate stems. But when I'm doing that, I want to be very clear in my field form that these might all comprise one genetic individual. And you'll see on your field forms that they ask you whether you have counted ramets or estimated how many genets you have. And a genet might consist of all of these plants here together and then separated over here, for example, by this group of plants, which might be a completely different genetic unit here. So in this case, I might say that I think I have 2 genets, but I will also report the number of ramets that I have within each genet. And as long as you're clear about the methods that you're using, you'll do just fine. But you want to be very clear so that other people who might return to the site years on end will be able to replicate your methods. And then we'll get a consistent idea of whether the population is expanding or declining. Okay, so now I have a rare plant that's actually quite common or dense in a particular area, so that there are a lot of stems potentially to count. And if it's widespread over an area, you may have to do some estimating and extrapolation. So what I do when I'm out in the field is I use what's called a quadrat, or basically a square. And you'll see I'm using really high-tech materials here. These are the reflectors that you put in your driveway to make sure that the plows don't completely clobber your driveway. They're very lightweight, they're very flexible, but they're also very tough, easy to carry around in the field. And I have 4 separate ones, and I've laid down the first 3 in a square, and I'm going to lay down this 4th one here in a square. And these are 4 feet by 4 feet. So it's important to know how much area you've actually got inside your quadrat. So I'm going to pretend that this rare plant, this grass-leaved euthymia, is the plant of interest that I'm going to try and count. Okay, so now I've laid down my quadrat, all right? I've got this 4 foot by 4 foot area, and I'm just simply going to count the number of stems, or ramets, of this grass-leaf goldenrod inside this quadrat. So I've got 1, 2, 3, 4, 5, 6, 7, 8 within this particular area. And once again, I don't know if these are all attached to each other. They may be ramets, all part of one genet, but I don't care. I'm simply counting stems to understand the density of these plants here. But soon we'll see that these plants vary in density over the entire area of this field. So now what do we do? Okay, so we're still dealing with our rare plant, but as you can see, there are a ton of stems in this particular location. So there is no prayer of actually counting all of these stems. Instead, what we're going to have to do is to estimate the number of stems by laying down some representative quadrats throughout this whole stand, maybe 10, maybe 20. And I'll count the number of stems within each of the quadrats, and I'll come out with a range of densities. And typically, we usually use the mean or the average density of the plants across all the quadrats to give a basic estimate of the overall density of the plants. So then what we're going to do is to map out the full extent of the population. So that's going to be our next step. Okay, so we are in an area that is incredibly dense and extensive coverage of the plant of interest. Now this could be a PCV's worst nightmare. Well, you've estimated throughout the population what the average density of the plants is. Now you need to get an estimate of the overall area that these plants occupy. And there's a couple of different ways that you can do this. The medium tech way to do it is if you happen to have a measuring tape and your population is small enough that you can actually measure out the boundaries of that population. So that's one possibility. A slightly higher tech possibility is to go to the farthest extents of the area that is occupied by this population and actually take a GPS reading at each of those corners, maybe north, south, east, and west, or the lowest boundary, the highest boundary. We're going to walk to the back boundary here, and by doing that, I actually get points that I can lay into a polygon. I can actually map the extent of the population. So that's sort of two methods of being able to estimate the area of the population. And finally, there's a pretty low-tech, but actually very effective method for actually measuring the extent of your population. It's called your feet. And what you want to do is to estimate the width of your stride when you're walking as you would normally be walking. So in my case, my stride is approximately 2 feet. So if I am walking from one end of my population to the other end of my population, I can measure the distance by the length and number of my strides. So I'll do that and walk basically a perimeter around my population so that I have a sense of the overall area that is occupied by these plants. So say I've paced out the length and width of the area occupied by my population. How do I estimate its actual area in square feet or preferably in square meters? Well, I can roughly estimate the area by assuming that my population occupies a circular plot or a rectangular plot or perhaps an ellipse, which is an elongated circle. I can use formulas for calculating areas that I learned from geometry way back in high school. For example, the area of a rectangle would be the length times the width. Or there are several online tools that can help me remember how to do that. Alternatively, I can use Google Earth Pro or ArcGIS if I have those tools on my computer. I can draw a polygon onto a map that approximates the boundaries of the area that I measured, then use tools in the software to calculate the area of that polygon. For our purposes, though, it's fine to estimate the area that your plants occupy based on a reasonable approximation of the shape of the overall population. So when you have maybe hundreds of plants that you need to count, it can be kind of hard to keep track of how many plants you've actually counted so far. I personally can get distracted, and so I love this little tool, which is a counter. You can get these from forestry suppliers or a number of different outlets. And all I have to do as I'm walking around the population and counting the plants that I see, I can simply click. And each time I do that, it logs the plant that I've just counted. And that way, I just establish an easy rhythm together with my stride, and I can keep track of the hundreds of plants that I might actually have to count. Okay, so now I've laid down another quadrat, and I'm going to count the number of plants that are in here to obtain a new density estimate. But I also want to make the point that when you're counting these plants, you want to distinguish between the plants that are actually reproducing, like this one, and the ones that are not reproducing. Because your field form will ask you how many plants you have and what types of plants you have. Are they immature? Are they mature but not reproducing? Or are they reproducing? And if they are they in flower? Are they in seed? Or are they in bud? So you definitely want to keep track of your reproductive and your non-reproductive stems.